Welcome back, everyone. Today we are wrapping up our discussion of enzyme mechanisms with a few more examples. Learning outcomes from today, we are continuing to learn how our groups can perform catalysis and the types of catalysis that we are seeing. We're seeing a lot of acid-base catalysis and some covalent catalysis as well. Today, we will see how cofactors can participate in the catalysis with a mechanism from carbonic anhydrase. Finally, we are going to kind of pull all of what we have learned together today and compare and contrast the many ways enzymes behave as catalysts. Today's Meet a Scientist moment is Kelly Chacon, who uses she, they pronouns. She is an associate professor of chemistry at Reed College. They are of white and Mexican descent. And they study metal ion transfer between proteins. So today is going to be the first time we really have a clear example of an enzyme that uses a metal ion to perform catalysis. And Kelly studies how metal ions are put into proteins, are transferred from one protein to the next. Cool stuff. So we are continuing our journey of learning about covalent catalysis today. We saw an example of this with serine proteases, but we're going to give it a bit more focus today. Sometimes covalent catalysis is called nucleophilic catalysis because there's some type of nucleophilic attack that's often performed when we're forming a covalent bond, right? This nucleophile is most often from the catalyst, and the electrophile is most often from the substrate. Here are some examples of nucleophiles that we would expect to see in biochemistry. So very common nucleophiles are going to look like hydroxyl groups, right? Or thiol. The OH and SH are more or less the same when it comes to the type of chemistry we'd expect. Of course, we should recognize that the nucleophilic form, though, is O-. It's a much stronger nucleophile than the OH. Same with that thiol. But it's not just this hydroxyl group or thiol that can do a nucleophilic attack. We, of course, can have the lone pair from amino groups or a lone pair on nitrogen within an imidazole group that we might see on a nucleophile like histidine. So nucleophiles that you'd expect from organic chemistry, again, we definitely see these in biochemistry, particularly OH, SH, and NH. The most common electrophile that we are going to see in biochemistry is going to be the thing that looks like a carbonyl carbon. Either it's a carbonyl, maybe it's an amine, maybe it's a metal ion. But for the sake of this class, really things that look like carbonyl carbons. What are some amino acids that we would expect to see performing this type of covalent catalysis? Well, histidine, cysteine, lysine, aspartate, and serine are reasonably nucleophilic and can form that transient covalent bond that then will be broken at the end of the mechanism and leave as a leaving group. Later in this class, we'll see examples of cofactors that also perform the same type of reaction: thymine pyrophosphate, pyridoxal phosphate. Don't worry too much about that now, but no, cofactors also do this type of chemistry.